Yes. Oh, second.
Sir, it's a common presentation. The session is brief.
May I?
Yeah, yes.
A very good morning and warm welcome to Miss Krutika Pardeshi, today's guest speaker, all my colleagues and the delegates who are attending this webinar on emerging trends in co-process excipients and architecting dosage forms, which is organized by Department of Pharmaceutics of Gokhale Education Societies, Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research, Nashik. We know that Gokhale Education Society is having a legacy of 105 years, having 140 plus institutes spread over three zones of Maharashtra, that is Mumbai, Palghar, and Nashik. We have a colleges, almost all are NAC accredited and re-accredited schools, and technical educations, all are ISO 9001 2015 certified. And Gokhale Education Society is known for providing quality education at very affordable cost and on a principle of above self and above politics. And we have a providing education to all from KG to PG, not only in urban areas but very rural area, mainly in Adivasi area. We have a lot of work there, and we are having a technical education institutes like engineering college, management college, then health education concern, physiotherapy college, nursing college, and this pharmacy college, which was established in 2012, completed 10 years of the college, and during these 10 years college has progressed many things. Initially intake was 60, now it becomes 100. Then we started diploma program in 2017, then we started M-Pharm in 2020 in pharmaceutics, then M-Pharm in quality assurance in 2022. And very importantly, we do have a research center affiliated to Savitribai Phule Pune University. There are many laurels brought by students and staff members to this college. Among that, I'm very proud to say that ours is the first college in India got accredited by NBA in 2019, and very happy to share with you today. Just yesterday, our college got accredited by NAC with the A grade with CGPA 3.22. This is because of support and contribution of all stakeholders, among that student, teachers, parents, then recruiters. Everybody has contributed a lot, and we are proud, and I'm very happy to share with that. Ours is the only pharmacy college in Nashik district got maximum CGPA 3.22 with A grade. So this is all about our college. Now we have been organizing a very unique kind of seminar, that is student and one of the industrial person, and all this we could do because of visionary leader late Sir Dr. M.S. Gosavi sir. He served for education industry near about 65 years, and he's having a mini records and he brought. But unfortunately, on 9th July 2023, it's a great loss for our society. Sir passed away on that day. And college is organizing very unique activity. That is, we have started a webinar series with, we are asking to one of our alumni who is doing well in the industry or academic, and another the person from industry, which can guide to the our existing students. So this is a very unique activity. And among this is the third webinar we organized in this academic year. And today we have a Miss Krutika Pardeshi, assistant professor at Sandip University, Nashik, and she is going to talk on architecting pharmaceutical dosage form. And we have Dr. Sita VG, assistant manager, technical services. I am Limited, Mumbai, Maharashtra, and around last two seminars we had around 700 registration from different corners of Maharashtra, and the same case today also we have a 500 plus registration for this webinar, and I'm very sure that this webinar could be very much useful for the existing students and those all delegates who are attending this seminar on emerging trends in co-process excipients and architecting dosage form. Once again, I welcome to both the speakers as well as all the delegates who are attending this seminar. Thank you. Thank you very much. Over to Krutika.
Thank you very much, sir. So shall I start with the session?
Wait a minute, Krutika. Wait
Just
a
a
minute.
minute, Krutika ma'am.
So today's webinar speaker one is Ms. Krutika H. Pardeshi, madam. Madam is working as an assistant professor, Department of Pharmaceutics, School of Pharmaceutical Sciences, Sandip University, Nashik. Madam has been awarded as prestigious alumni of Gokhale Education Society, Nashik, on centenary year by the hands of Honorable Raghunath Mashilkar sir in 2018. Professional experience of Madam. Madam is currently working as an assistant professor in Gokhale Education Society, Sir Dr. M.S. Gosavi College of Pharmaceutical Education and Research, Nashik, since August 2018. Now, currently, Madam is working as an assistant professor in Sandip University. Publications. Madam has published papers. The research article on preparation of spherical crystal agglomerate of arithmether using crystalloco agglomeration technique in International Journal of Pharmaceutical Science. Madam has also published review article on current scenario in diagnosis of COVID-19 in World Journal of Pharmacy and Pharmaceutical Sciences. Madam has published a book entitled Computer Application in Pharmacy Practical Manual, and Madam has also published a book entitled A Textbook of Pharmaceutical Microbiology. We heartily welcome you, ma'am, for the webinar. Over to you, Krutika, ma'am.
Thank you so much, ma'am. Am I audible?
Yes, madam, you are audible.
Yes. Can you please share my slides so that I can start? Yes. My screen is visible?
Yes, ma'am. Your
Yes,
screen
yes,
is visible.
it's visible.
Yes. Okay. Thank you so much. So before starting with the session, I would like to thank you, Principal Dr. S.V. Amrutkar sir, for giving me the opportunity to interact with the students of my college, that is Sir Dr. M.S. Gosavi College of Pharmaceutical Education and Research. I'm very happy that I have again connected with my institute from where I have started the journey of the pharmacy. And I would like to congratulate the whole team of the MSD Copper for getting the accreditation from the NAC. Heartiest congratulations to all of you. And now let's move toward the topic. I'm going to talk on the architecting pharmaceutical dosage form. This means how to design a pharmaceutical dosage form. So before seeing how to design the dosage form, let's see what is the dosage form first. A dosage form is defined as a physical form of drug that may be a solid, liquid, or gas, which it is delivered in its proper form to the particular site of the action within the body. Now, dosage form is basically a composition of the API, that is active pharmaceutical ingredient, and the excipients, which is composed of a 90% of the dosage form, which are required for the various purpose that I'm going to discuss later. So these dosage form are prepared with the aim that they have to be targeted to the proper site where it will show its pharmacological action. Transforming a drug into proper dosage form not only provide the advantage like the handling, ease of administration, but also improve the dissolution and bioavailability of the various drugs, which shows the low bioavailability or the low dissolution rate. When we are thinking about the designing of the proper dosage form, physical, chemical, and biological properties are there that need to be considered of the drug moieties and also the excipients that we are using in the designing of the dosage form. Now, what are the pharmaceutical excipients? Now, excipients are the other materials than the drug, but they have the various purpose, though they are intact, they do not have any pharmacological action. Pharmaceutical excipients are the substance other than API that have been appropriately evaluated for the safety and intentionally included in the drug delivery system. Now, pharmaceutical excipients can increase the bulk of the dosage form, can improve its dissolution, can help in compressing, can help in increasing the bioavailability, and many more advantages are there for the pharmaceutical excipients. If we talk about the ideal properties of the excipients while designing in the dosage form, we should focus on the following points. The excipients we are selecting for the dosage form should be stable with the drug and inside the body also, and it should be reproducible. It should not interact with the drug molecule, and it should be pharmacologically inert, that it do not possess any pharmacological activity. It will only help the drug moiety to increase its pharmacological action. It should have a desired functionality and it should be cost effective. If we talk about the functionality, there are various excipients which have more than one function. Later, I'm going to discuss about the multifunctional excipients though. Now, in designing of the dosage form, why these excipients are added? What is the role of the excipient? Excipients improve the integrity of the drug product during storage. For example, if any drug is hygroscopic in nature, if we add a few excipients which will reduce its hygroscopicity, obviously we are going to increase the stability and the storage of the excipients. Most of the excipients are assist in effectiveness or the delivery of drug in use. Like for example, if there is a potent drug whose dose is around 10 mg, so it is not possible to take that drug alone. We need to add some excipients to create a bulk so that it can be easily administered. It also protects, support, and enhance the stability, bioavailability, and patient acceptability. Now, for example, if any drug is bitter in taste, if any drug is not palatable or not having patient acceptability, we can add few excipients which will mask the bitter taste of the drug, which will coat the drug, and thereby we can administer the drug and improve its stability also. It also aid in processing of the drug delivery system during the manufacturing process. Like for example, increasing in the compressibility, increasing in the dissolution, or providing a suspending characteristics in order to prepare a suspension. It also assists in product identification and also enhance the overall safety when we are creating a dosage form with the proper excipients. Now talking about the dosage form design. Why there is necessity to design a dosage form? It is very easy. It would be very easy if we administer the drug directly. Why don't we take a paracetamol directly with the help of spoon? Or why don't we administer a drug directly to the systemic circulation? There are few points which helps us to identify that why this dosage form design is necessary. First point is to protect the drug. As I've told you, there are many of the drug which gets decomposed when they are stored at a certain temperature. They are stored at a lowest temperature. They should be protected from the atmospheric oxygen. They should be protected from the humidity because it may change its chemical form and decrease its pharmacological action. If we design a dosage form with the suitable addition of the excipients, it will protect the drug substance from getting influence with the humidity or atmospheric oxygen. For example, the coated tablets or the sealed ampules so that they can be protected from the environment. Next is the protection of the drug from the destructive influence of the gastric acid. Now, majority of the drugs are administered through the oral route. For example, the entry coated tablets. This consists of excipients like HPMC or polyvinyl acetate phthalate, cellulose acetate phthalate. With the help of these excipients, the drug has been coated and it protects the drug from the gastric acid and thereby protecting its originality, protecting its pharmacological action. Next is to control the bitter salty taste. Here also we can incorporate the taste masking agent or the flavors if we are preparing the pediatric formulations to provide the liquid preparations of the substance either in soluble or unstable form. For example, the suspension, if few drugs are not properly dissolved in a solution, it do not form solution, so we can go for the suspension and emulsion also by adding the suspending agents or emulsifying agents to provide a clear liquid dosage form, obviously to increase its solubility so that it can be easily dissolved, easily absorbed from the body, to provide rate controlling drug action. Now, there are various excipients which can be used in sustained release dosage form, extended release dosage form, in a capsules or the suspension, which release the drug at the control rate so as to decrease the patient's ability to take the dosage form. To provide optimal drug action from the topical administration, like for example ointment, waxes have been added which improves its property to application. It will help to apply the ointment properly on the skin. It increases the spreadability of the ointment. Creams, we are adding W by O or O by W emulsifying agent so that the creams will prepare with a proper emulsion. It can be remain stable for a longer period of time. If we want to transfer, if we want to administer the drug through the rectal or vaginal route, it is necessary that it should be formulated in the form of the suppositories. So it would be easy to administer the drug through this route. If we are directly adding or if we are administering the drug directly into the body circulation or the bloodstream, it is necessary that it should be completely dissolved, or if it is formed in the suspension, it should be properly suspended so as to avoid the clogging of the drug in the bloodstream. And last is the optimal drug action through inhalation therapy. In aerosols, propellant, we are adding few excipients also in order to improve the administration ability of the dosage form in inhalation therapy. Now, there are few consideration while we are designing a dosage form. It is not only about the excipients but about the drug and about the compatibility of the drug along with the excipients. Biopharmaceutical consideration, drug factors, and therapeutic consideration are the three major points that we have to consider whenever we are designing a dosage form. Now, biopharmaceutical consideration include what would be the absorption, distribution, metabolism, and elimination of the drug after designing a dosage form when we are administering it the different route. For example, paracetamol when incorporated into a tablet dosage form require much time to go into the systemic circulation as compared to the paracetamol injection. Drug factor include what are the physical and chemical properties of the drug substances. Therapeutic consideration include consideration of the clinical indications to be treated and a patient factor also, like the compatibility or the acceptability of the patients. So yes, biopharmaceutical consideration, we have to first look at what would be the route of administration for any kind of drug when we are designing a dosage form. Like if we want to go for oral route, it must be assured that drug should be properly solubilized, drug should be properly distributed. Then we have to take into the consideration about the metabolism also, how much amount of drug going into the systemic circulation. When we are designing a topical dosage form, the spreadability of the dosage form should be proper. When we are going with the parenteral dosage form, the suspension or the particle size of the drug is very important. For inhalation, the propellant and the drug which we are using, that should be have a particular particle size. Determining the pharmacokinetics like ADME, the drug candidate pharmacokinetic profile and the type of illness is important. Like for example, if the patient is suffering from a fever and he is unconscious and not able to take the medication through oral route, the injection route is most preferred. Route of administration is also considered, which comes under a therapeutic consideration. Now, what is the nature of the clinical indication? What kind of disease is there? And what kind of route of administration to be selected is necessary? If we are giving any of the dosage form which is required to be give the action within 5 minutes or 10 minutes, we cannot go for the oral route. We have to go with the injection route only or parenteral route only. In case of emergency, some of the nitroglycerin and loratadine antihypertensive sublingual tablets are available or the injections can be given. When we are talking about the age of the patient, like geriatric patients and the infants, they have a problem in swallowing the dosage form. Like liquid drops can be given or the syrup can be given, or nowadays many research is going on the mini tablets production, so that the infants or children can easily swallow that mini tablets. While designing a dosage form for the infants or the children's, we have to incorporate some flavoring agents or taste masking agents so that it can be easily acceptable by the children and they can take the medication easily. The drug factor include all the pre-formulation study parameters, starting from the organoleptic properties, bulk characteristics, analysis of the solubility and stability. We have to check that what is the solubility of the drug, what is the color odor of the drug, what is the taste of the drug, what is the solid state like amorphous or crystalline. Based on that, we can design the suitable dosage form. If the drug is potent, like for example, there are some drugs like domperidone is there, whose dose is around 10 mg. Nitroglycerin is there, whose dose is almost zero point 0.3 mg. And other dosage form, they are very potent and very less quantity. So we have to incorporate some bulk forming excipient so that we can increase its size, we can increase the mass so that it can be easily administered. Flow property, density, compressibility are important while we are preparing a tablet dosage form. Polymorphism includes various forms of the drug showing the different characteristics. Example could be a theobroma oil who are having more than 12 forms, but from that beta form is majorly used. When we talk about the solubility analysis, it is a known factor that drug should be soluble completely in order to get absorbed in proper amount. So before designing of the dosage form, the solubility analysis and stability analysis should be done. Now move towards the designing of the dosage forms that are novel nowadays. We will focus on what are the different dosage forms that are novel and how they are designed. What is the difference between the existing dosage form and the novel dosage form? Here I have taken few of the examples of the novel dosage form and going to discuss about how this dosage forms can helps in designing, means what are the different types of excipients, what can be added to design this dosage form. So very first is the phytosome. It is somewhat related to the vesicular drug delivery system. Phyto means related to the plants and somes related to the something which is covered over the surface. When we see the difference between the phytosomes and liposomes, a structural difference include in liposomes, the drug is incorporated within the core, or we can say the cavity, or sometimes it can be present in the layer also. Here you can see in the liposomes, API is distributed in the medium containing the cavity. When we are talking about the phytosome, API, active pharmaceutical ingredient, which is a phytoconstituent obtained from the plant, is the integral part, being the molecule stable with the help of hydrogen bonding to the polar head. You can see in the phytosomes, in the polar head, drug has been incorporated. What are phytosomes? I've told you that phytosomes are something which are incorporating the phytoconstituent and little bit structurally similar to the liposomes. These are advanced drug delivery system most popularly used to deliver the phytoconstituents or the herbal product in vesicular encapsulated form. A major component of this drug delivery system is phospholipid molecule. Now, there are multiple examples of the phospholipid molecules, including the synthetic and semi-synthetic agents that can be incorporated in the phospholipid molecule. This phospholipid act as an effective emulsifier and also have the access through the biomembrane. These phytosomes are preferred because it has a good bioavailability, it has the absorption property, and it is excellent therapeutic agent when we are compared with the liposomes because it consists of a hydrophilic head. It increases the hydrophilicity and helps to target the drug molecule by increasing the solubility and combining with the water molecule. Why there is need of the phytosome, why the phytosomes are incorporated or considered or preferred over the liposomal formulation. It enhances the absorption of lipid insoluble polar phytoconstituent through oral as well as topical route. It also improves the bioavailability. Now, when we are talk about the liposome and phytosomes, the phytosomal absorption is more because it consists of a polar phytoconstituent, and when we are formulating a topical route, it has a better availability as compared to the liposome. They are stable and cost effective because it consists of a phytoconstituents, for example, phosphatidylcholine. Now, an interesting fact is that a phosphatidylcholine is the excipients in the formulation of phytosome, but it can be act as an hepatoprotective agent when we are adding it in the formulation of the phytosome, hence produce the synergistic effect. Phytosomes can be used for the systemic targeting and it is widely used in cosmetic technology. Like, for example, cocoa husk phytosomes have been prepared by one of the scientists, which is a tyrosine kinase inhibitor, which inhibits the formation of tyrosine kinase and act as an anti brightening agent or a skin whitening agent. It helps with the entrapment efficiency when we are formulating a phytosomes with the phosphatidylcholine. The entrapment efficiency of the drug or cocoa husk phytosome increased up to 90%, and the skin whitening ability was improved on an average about 199.98 ppm. Now, what are the phytosomal formulation? Which type of formulation in which phytosomes can be incorporated? A soft gelatin capsule in which we can disperse the oil in the gelatin shell. We can formulate the phytosomes into hard gelatin capsules. We can formulate the topical dosage form where the topical dosage form can be formed with the help of forming a complex emulsion by dispersing a phospholipid complex with the liquid phase, and we can add it to the already prepared emulsion so that it can be stable. We can also go for compressing the tablets with the phytosomal formulation. What are the formulations or how they are prepared? Now, there are two types of agents, natural or the synthetic phospholipid. Now, natural may include the soya bean, then egg yolk is there, sunflower oil is there, which can be used as a natural phytosomal formulation. Or synthetic phospholipid may include phosphatidylcholine, phosphatidylethanolamine, and phosphatidylserine. We have to incorporate the one molecule of phosphoconstituent and one molecule with the synthetic phospholipid. This ratio can be 1 as to 1 or 1 as to 2. We can prepare it with the help of aprotic solvents such as acetone. The complex thus formed can be extracted with the help of lyophilization, precipitation, or the formation with the aliphatic hydrocarbon. Next is the gold nanoparticle. Now, nanoparticle is the colloidal drug delivery system. It forms the colloidal particles with 10 to 1000 nanometers. They are synthetic and semi-synthetic polymers that can be incorporated in the nanoparticular system. Now there are number of nanoparticles that are available, including the polymeric nanoparticles, micelles are there, dendrimers are there, solid lipid nanoparticles are there, magnetic nanoparticles include the gold nanoparticles and iron oxide nanoparticles, and micelles are also there. When the gold nanoparticles we are talking about, they are available in the different forms, starting from the nanosphere, nanoshell, nanorod, clusters, cages, and nanostar. These are the nanoparticles which can be incorporate a very small drug molecules or the biomolecules also, like DNA, RNA, and the proteins. They are more stable, they are non-toxic and easily synthesized because it do not require harsh excipients. It only requires a simple form of the excipients. How the synthesis of the gold nanoparticle is done. Now there is a well-known method of the top-down and bottom-up approach by which we can synthesize the gold nanoparticles. Top-down process involve the crushing of the bulk material into smaller nanoparticle, whereas bottom-up approach involve synthesis of the nanoparticles starting from the atomic level. Now there are two stages by which we can formulate the gold nanoparticles. In first stage, we have to identify the gold precursor and we have to prepare an aqueous gold salt solution. It can be reduced with few reducing agents like citrate, and the stabilization of the gold nanoparticle is done with the help of capping agents. Now, an interesting fact that when the gold nanoparticles have been discovered, they have been used by the scientists to treat the cancers or the cancerous cells or the breast cancer. Gold nanoparticles are the most stable dosage form or the metallic nanoparticles. A capping agents and the stabilizing agents are the two important excipients while we are considering the formulation of the gold nanoparticles, because stabilizing agents help to prevent the agglomeration of the gold nanoparticles, whereas capping agents are important again for reducing the nanoparticles to grow further. They are amphiphilic molecules, for example, polyethylene glycol is there, PVP is there, and EDTAs are there. Now, there are three methods by which we can synthesize the gold nanoparticles: a Turkish method, the burst method, seed mediated method. One of the newly formed is the digestive ripening and biological synthesis of the gold nanoparticles. Just a minute, sorry. Now the first method is Turkish method. AUNPs prepared using this method have a size range of 1 to 2 nanometers. The basic principle of this technique involved the reduction of the gold ions using some reducing agents like amino acid, ascorbic acid, and the UV light. The stabilization of AUNPs can be done with the help of PEG, PVP, EDTA, which will help to stabilize this agent. Next is the burst method. Here, the bursting of the gold salt from organic solvent to the aqueous solvent can be done with the help of some agents like tetraoctyl ammonium bromide. Seed mediated method is there in which the seed is transferred from the metal salt with the help of weak reducing agent like ascorbic acid. Digestive ripening is the very convenient method. We just have to heat the colloidal suspension at very high temperature for 2 minutes and thereby reducing the temperature at 110 degrees Celsius for 5 hours using alkane thiol. Temperature is the major factor for determining the size distribution. If we continued at a heating for 5 hours at a particular temperature, it will decide the gold nanoparticles size range. Now, biological synthesis is nowadays very useful for the synthesis of the gold nanoparticles because it reduces the use of very harsh chemicals, heating, etc. This biological synthesis is very clean method, biocompatible method as compared to the chemical synthesis. Some of the bacteria like fungi, eukaryotes can be used for the synthesis of the gold nanoparticles. Numerous biocomponents present in plants like flavonoids, phytosterol, quinines, etc. play an important role in the synthesis of AUNP because it consists of a different types of flavonoid. It speeds up the reactions and stabilization of the nanoparticles. When we are talking about the bacteria consists of a negative cell wall and it can be easily incorporated the gold nanoparticles which are positively synthesized and thereby they're increasing the surface area or the capability of the formulation or entrapping ability of the dosage form of the gold nanoparticle. Now, the stabilization of nanoparticle is the important technique after synthesis of the nanoparticle. Nanoparticle can be stabilized with the help of various agents like sodium bromide, sodium hydride, sodium citrate. Why this stabilization is necessary? In order to avoid the agglomeration that happens because of the repulsive forces. During the chemical synthesis, these capping agents or the stabilizing agents are added. However, during the biological synthesis, stabilization of nanoparticle can be successfully achieved using the biomaterial rich in antioxidant. Next is the liquid solid dosage form. This is also one of the new concept in which the liquids are converted into a solid form and liquid is also present inside the dosage form. A liquid solid system refers to the formulation by conversion of liquid drugs, drug suspension or drug solution in non-volatile solvent into a dry, non-adherent, free-flowing compressible powder. This can be done by blending the suspension or solution with the selected carriers and the coating material. How this liquid solid dosage form is prepared. The drug is dissolved in an organic solvent followed by soaking of the solution by the high surface area like silica, and the binding of drug is happened due to the drug particle gets agglomerated to the carrier. The technique of liquid solid compact and liquid solid microsystem is new and promising approach towards the dissolution enhancement. Now there are two major components in the formulation of the liquid solid dosage form. First is the coating material or the carrying material and second is the non-volatile solvent. Coating material forms a uniform film around carrier particle. It helps the particle aggregation, improves the flowability by covering the wet particle which absorb the excess liquid. Example of a well-known coating material is Aerosil 200. Non-volatile solvent has a capability to dissolve the drug adequately. It is miscible with water having increased boiling point like propylene glycol and fixed oil can be used as a non-volatile solvents. How the drug gets released from the liquid solid dosage form? By increasing the surface area of the drug, by increasing its aqueous solubility because we have incorporated a non-volatile solvent into the preparation, improve drug particle wettability due to the co-solvent effect of the used vehicle. Next is the self nano emulsifying drug delivery system, SNEDS. It is a nano-emulsion pre-concentrate anhydrous form of the emulsion. Nano-emulsions are homogeneous liquid mixtures, and the major components in SAID includes the oil, surfactant, and co-surfactant that can be emulsifier or solubilizer. What do you mean by self nano emulsifying? We have to prepare the emulsion, and when a GI motility that is naturally occurring in the body help in agitation. When this agitation is done, approximately 200 nanometer or less than a size nano emulsion has been prepared inside the body. Now, co-emulsifier has been added, which can facilitate the formation of the nano emulsion. Compared to the other types of nano emulsions, SNEDs are having a long-term storage, have a physical chemical stability. It can be filled into the various kind of dosage form, which is not in the case of the other types of nano emulsions. It can be incorporated into the tablet, into the capsule, or various dosage form, so that we can increase the stability also, we can increase the patient acceptability also. Now, why the SNEDs are important? SNEDs are important is first because it has a long-term colloidal stability as compared to the other dosage form. If we are formulated the SNEDs with the optimum conditions. Second, it has the ability to solubilize both the forms of the therapeutic agents like hydrophilic also and hydrophobic also. In case of hydrophilic drug, W by O emulsion can be prepared. In case of hydrophobic drug, O by W emulsion can be prepared. It can be helped to improve the stability of the therapeutic agent. Like for example, there is one drug, cefodoxime paclitaxel, which found that the stability of this drug is improved because it has been encapsulated and formulated as a SNED. It also has a greater aesthetic appeal and skin feel. Now, SNEDs have been incorporated in majority of the phytoconstituent and the cosmeceutical preparations. Because of the transparent nature, translucent nature, it gives a greater aesthetic feel. It can be easily applied. Aerosol of spray formulation, roll-on formulations can be prepared with the help of SNEDs. It has the improved dermal and mucosal transport because it has the improved the bioavailability of the many drugs, which may include few of the example is curcumin, ramipril, and enzalutamide. Three major components are there as we have seen: oil phase, surfactant, and the co-solvent. Oil phase include maximum solubilized capacity. When we are selecting the oil for the formulation of the SNED, we have to consider the solubility of the drug into the oil phase. Because it is a functional parameter, it is a key factor which influence both in the formulation and the loading capacity of the drug absorption. Various types of oils like castor oil, soybean oil, coconut oil can be used as an oil phase. They exhibit relatively low drug loading capacity and poor emulsification efficacy because they are naturally irritable oils. We can incorporate the modified medium chain or long chain triglycerides. Few of the example may include capriol, Captex, Picol, which are most commonly employed oil phase, which have a maximum drug loading capacity, and it can be also increase the absorption of the drug. Medium chain triglycerides are preferred because it has a better solubility and it can help SNEDs to get self emulsified properly. Next is the surfactant. The surfactants whose HLB scale is more than 12 are mostly preferred. For example, polysorbates, castor oil esters, polyglycerides. While taking into the consideration of the factor of the surfactant, concentration of the surfactant is very important because it can affect the formation of the particle size and also cause a concentration depending toxicity. Next is the co-solvent. Now this co-solvent are very important because It usually required with the help of another surfactant because one surfactant cannot reduce the interfacial tension. The purpose of adding the co-solvent could be the increasing in the drug loading capacity or to change the self-emulsification time like we do in modified drug release or sustained release, and to modulate to increase or decrease the droplet size. Various types of co-solvents can be commonly used, like includes the propylene glycol, polyethylene glycol, and a newer co-solvent, transcutol, can be used in formulation of the SNEDs. Now, how this formulation takes place or how the SNEDs can be formulated, these are the methods similar to the formulations of other nano emulsions, which may include the high energy and low energy emulsification, which includes the ultrasonication, high pressure homogenization, phase inversion, solvent displacement, phase inversion, high pressure homogenization methods. Now, we have discussed about the different novel formulations and their excipient role in the formulation of this novel dosage form. What are the excipients, why they are selected, how they are increasing or decreasing the palatability or the acceptability of the dosage form or increasing the solubility. Now, we'll see the role of some functional excipients in the dosage form design. Now, what do you mean by the functional excipients? Functional excipients have a function to improve the bioavailability, to improve the dissolution, and many more. Means these excipients have a multifunctional role. For example, if one excipient is added to increase the bulk, it will improve the dissolution also. According to the International Pharmaceutical Excipient Council, excipients can be divided into new and established chemical excipients. New chemical excipients can be classified into modified, means the excipients whose chemical structure is been modified, thereby improving its efficacy. Co-process excipients mean two or three of the excipients have been formed together, co-processed, and the novel excipients whose ability or being an excipient has been newly discovered. Multifunctional excipients are the excipients with the multiple roles in the dosage form. For example, direct compressible filler can also act as a binder or disintegrating agents. Poor physicochemical properties, low aqueous solubility, and membrane permeability can be improved if we use a multifunctional excipients. There is various research material available in which you can see that with the help of co-process excipients, with the help of multifunctional excipients, the major problem that arises in the designing of the dosage form includes its poor physical chemical properties, thereby decreasing its compressibility, solubility, membrane permeability, absorption can be overcome if we incorporate a co-process excipients. Now, these are some case studies with the newly designed excipients which have been found that there is improvement in the dosage form. First is the formation of inclusion with the cyclodextrin. Now, cyclodextrin form a solid liquid dispersion. When a cyclodextrin as a functional excipient is used in one of the study of the carbamazepine, it was found that in presence of a cyclodextrin complex with HPMC, 95% of the solubility increase when compared to the normal dosage form. Similarly, when the ODTs, orodispersible tablets, have been prepared by one of the methods, including the solid dispersion with the help of beta cyclodextrin and the drug eslicarbamazepine, 100% dissolution was achieved within the 60 minutes compared to the normal commercially available tablet formulation in which only 72% of the dissolution took place. It indicates that formulation of the solid dispersion with the help of cyclodextrin can improve the solubility, can improve the dissolution as well. Now there are few disintegrants like sodium starch glycolate and cross-providin. When combined with the two of the valsartan and irbesartan antihypertensive drug, it was found that the fast disintegration was observed. Like for example, disintegrated within 2 minutes in case of the valsartan, and dissolution of the irbesartan is also improved when we have incorporated the 5% of the jackfruit starch as compared to the dissolution of pure drug, which is only 43%. Means only after incorporating the 5% of the starch, the dissolution rate was improved from 43% to the 60%. Next are the pH adjustment excipients. They modulate the microenvironmental pH, changing the drug crystallinity and providing a non-favorable nano-emulsifying forming environment. The combination of Na2CO3, which is an alkalizing agent, and one of the polymer, that is poloxamer 47, when they are combined with the aciclofenac, which is an NSAID, result into the increase in vitro dissolution. And also this was attributed because of the three mechanisms that include: it changes the pH in the acidic level, changes the crystallinity of the drug, and it provides a suitable environment where the nanoemulsion can be formed. Fourth is the amorphous solid dispersion. Now, preparing of the solid dispersion with the help of PVP, HPMC, HPMCP, that is hydroxypropyl methyl cellulose, chitosan, converting the drug into the amorphous stage, improve the solubility and the bioavailability. We are aware about the fact that the solubility and the bioavailability of the amorphous drug is more as compared to the crystalline state. So we can convert the crystalline drug to the amorphous dosage form by incorporating it with the HPMC or sodium alginate or sodium starch glycolate. Next is the sugar. Now, how the sugar can be act as an excipient? We are aware that the sugar can be added only to mask the taste of the bitter drug, or it can also help to give the most patient palatability. Mannitol is one of the form of the sugar when combined with the ketoprofen by the technique of melt, dispersion and kneading method. It was found that the dissolution rate of the ketoprofen was increased as compared to the plain ketoprofen dosage form. Next is the filler materials. Now, microcrystalline cellulose and the lactose are the two major excipients which can be added as a filler materials, but it has the multifunctional ability to increase the dissolution. One of the research carried out with the help of antioxidant quercetin when combined with the microcrystalline cellulose with the help of the crystallization technique, fluid braid crystallization, it was found that the antioxidant effect of the quercetin was increased and dissolution of the drug was also found to be increased. Now, next is the lactose, which is very, very well known insoluble filler material. All the formulation containing functional excipients revealed enhanced dissolution and drug release from the nanodispersion overall during the 2 hours evaluation. Now, I have taken the example of the carbapol and explaining with the patient-centric designing of the dosage form. Now, what do you mean by the patient-centric designing of the dosage form? Patient-centric means we have to take into the consideration about the need of the patient. Like two example, if we have considered, the patient would ask, how many times I'm supposed to take this medication? Or do you have a medication in a smaller size? Now, majority of drug though have a little amount of the dosage form or they are in mg concentration. Few of the drugs are there whose concentration is around 500 to 1000 mg. Along with that, if we want to incorporate the excipients which will improve its solubility, dissolution, increase its compressibility, or providing the suitable reducing in the agglomeration rate, that formulation will be increased and tablet side would be more, and it will not be accepted, not be consumable by the patients, maybe a geriatric or pediatric patient. So carbapol is one of the newly formed or one of the well-known excipients which was used in the metformin tablet by a Lubrizol company along with the different forms of the carbapol. The drug loading capacity was improved to 80%, which reduces the size of the tablet, means they have prepared the 22% smaller tablet size. So it can be easily accepted by the patient, it can be easily administered to the patient and do not have any problem in the swallowing. Last is the frequency of the dosage form. Many a times patient forgot to take the dosage form if they are prescribed 2 times a day or 3 times a day, which is very difficult for everyone to consume the dosage form. So there are many research going on to reduce the frequency of the dosage form. Once a daily sustained release tablet could be the one of the example. A well-known cold or flu medicine, Mucinex, helps to in that a carbapol is added enable the generation of unique bilayer tablet, and it has been utilized to show the release of the sustained or extended release so that it reduces the dosing frequency. The guanifencin is the drug which is present in the Mucinex, and it has a challenge because it is a water soluble drug and it would be very difficult to release to control the release of this drug. When carbapol has been incorporated with the help of this drug, high drug loading and good control can be possible when we are adding with the water soluble API. So carbapol has been discovered that can be added with the water soluble API, and that will improve or increase the extended release, thereby decreasing the dosing frequency. So these are the few examples of the excipients in the designing of the dosage form, why the excipients are important or the key role factors for the designing of the dosage form. Details about this excipients and the co-process excipients will be explained by the expert Dr. Sita in next session. So with this, I conclude my session in the designing of the dosage form. Thank you very much for giving me the opportunity to explain the topic designing of the dosage form. MSG Copper team, over to you. Thank you so much.
Thank you, ma'am. Delegates having questions, you can paste your questions into the chat box.
Yes, I'm able to see one question. In chemotherapy, gold nanoparticle formulation can be prepared. Yes, because gold nanoparticles, when they have been discovered, they are used for the determination or the identification of the cancerous cells. So yes, it can be prepared for the formulation to treat the cancerous patient in the chemotherapy.
Any more questions?
As there is no question, we can see in chat box. We have explained and see.
Okay, as there are no more questions, thank you, ma'am. Thank you for our prestigious alumni of a college, Ms. Krutika H. Pardeshi, for giving insights on different aspects of formulation of pharmaceutical dosage form, role of excipients in formulating novel dosage forms. Ma'am has also emphasized on formulation of gold particles, which are being widely employed in biotechnology based on their unique properties and multiple surface functionalities. Ma'am has also emphasized on self-nano emulsifying drug delivery systems, which are one of the emerging strategies developed to tackle the issues associated with their oral delivery. Thank you so much, ma'am. Thank you once again.
Thank you so much, ma'am, for giving me an opportunity to interact with all of the students of my college, that is MS Gosavi College. And thank you so much for introducing me. It would be I'm very happy to be with all of you. Thank you so much.
Thank you so much.